For 3 billion years, life on Earth was blind. There were no eyes, not one. No creature on the planet, anywhere, could see another. No predator tracked its prey by sight. No prey fled from a passing shadow. No animal could recognize another animal across a distance. The oceans were populated, vastly populated with microbes, with soft-bodied things of many kinds. But everything that lived in them lived in a world of chemistry and touch, a world of scent in the water, a world of currents and pressure waves, and the soft bump of one body against another in the dark. Somewhere around 530 million years ago, in a span of geological time, brief enough that we can resolve it to within a few million years, that changed. Light became information. A spot on the surface of an early animal became sensitive to the difference between bright and dim. The spot became a dimple. The dimple became a cup. The cup acquired a clear covering. The covering focused. The focus sharpened. And within what may have been only a few 100,000 years, a blink in the time scale of the planet, the first creature on Earth opened the first real eye and looked at the world. Every animal alive today, every fish, every insect, every snail, every mammal, every bird, every human inherited its ability to see from that one brief, astonishing transition. The eyes you are using to read this, or the ears through which you are listening to it spoken, trace an unbroken chain of development back to the strange sea creatures of the early Cambrian. This is their story. In a deep and almost uncomfortable sense, your own. Before the Cambrian, there was the Ediacaran. It is the name of a period of Earth's history that runs from roughly 635 million years ago, just after the last of the great snowball Earth glaciations, down to the base of the Cambrian at 538 million years ago. And it is the name of a world almost completely unlike anything we would recognize. The oceans of the Ediacaran were warm, shallow, and quiet. The polar regions, no longer fully iced, were comparatively mild. The atmosphere, after the final pulses of the Neoproterozoic oxygenation event, had reached levels of atmospheric oxygen that could support large, soft-bodied multicellular animals for the first time. And on the shallow sea floors of the late Ediacaran, those animals had arrived. We call them collectively the Ediacaran biota. They are preserved most spectacularly in South Australia, in the Flinders Ranges, particularly in a place called Ediacara Hills, from which the whole period takes its name. They are preserved also on the coasts of Newfoundland, in the Russian White Sea region, and in scattered sites on every continent. They are, as macroscopic organisms, the first large creatures the Earth ever produced in any abundance. But they are strange. They are deeply, almost unsettlingly strange. The Ediacaran biota include frond-shaped organisms rooted to the sea floor, reaching up into the water like soft, feathered sea pens. It includes disc-shaped creatures, some a few centimeters across, some nearly a meter, lying flat on the sediment. It includes ribbon-like bodies, segmented with rippling patterns, of unclear orientation and unclear feeding habits. It includes creatures that look vaguely like flat quilted mats, showing repeating geometric patterns of small rectangular panels. Creatures whose entire body plan has no equivalent in anything alive today. Dickinsonia, one of the most famous, was an oval body up to a meter long, segmented along a central axis, probably capable of slow movement across the sea floor. What did they eat? Almost none of them had mouths, not in the sense you would recognize. The majority appear to have been osmotrophs, creatures that absorbed dissolved organic molecules directly from surrounding water across their body surfaces. Some may have hosted internal symbiotic bacteria that produced food for them, in the manner of modern chemosynthetic communities. Some may have filter-fed on very small particles. From any of the evidence we have, were predators. None of them had teeth. None of them had claws or spines or armor. And this is the key point, had eyes. The Ediacaran Ocean was a garden. It was a garden in the sense that modern botanical gardens are gardens, a space of slow growth, of extended bodies anchored in place, of patient metabolism, of gentle filtering. There were movements in the Ediacaran seas. There were slow crawls of Dickinsonia-like animals across microbial mats. There were the drift of the fronds in gentle currents. There were the growth and decay of individual creatures across their lifetimes. But there was nothing, as far as any of the evidence shows, resembling a predator. Nothing that hunted, nothing that fled. This is the world that was about to end. To understand how radical the Cambrian arrival of eyes was, you have to understand what was missing before them. Eyes in an animal body do one very specific thing. They convert patterns of light into patterns of information about the world at a distance. An animal has to rely on other senses. It can taste the chemistry of the water around it. It can feel pressure waves through receptors in its skin. It can touch what it encounters directly. It can, in some cases, detect low-frequency vibrations transmitted through the substrate. But all of these senses are, in a fundamental way, short-range. They give you information about what is near you, or what is in direct contact, or what is recently past. They do not give you a detailed picture of what is over there, across the water, around the corner of the rock. This means that a world without eyes is a world organized around very different dynamics than the ones we are used to. In a visual world, the world you live in, a predator can see its prey from a distance and can begin a pursuit before it is committed to any particular direction. A prey animal can see a predator approaching and can flee, taking an evasive path calculated against the predator's position in real time. Individuals of the same species can recognize each other across a crowd, can track potential mates or rivals, can interpret posture and display and coloration. An entire economy of communication and competition is available, made possible by the ability to receive detailed, structured information about the world. In a non-visual world, the world of the Ediacaran and everything before it, none of this is possible. A predator hunting by smell finds its prey only when the chemical trail is strong enough, meaning typically after the prey is already wounded or dying. A prey animal fleeing by touch flees only after it has been touched, meaning typically after the predator has already begun its attack. Individuals of the same species may recognize each other only by pheromone release, only in close proximity. Communication is reduced to chemistry, and chemistry is slow and local and blurry. The consequences for ecological dynamics are enormous. Without vision, the pursuit and evasion arms race that dominates modern ecology simply cannot run. Without vision, armor and spines and speed confer much smaller benefits. You are not being targeted across a distance, so the value of being difficult to target is limited. Without vision, organisms tend to be soft, slow, and sessile. They do not need to move fast because nothing is coming at them fast. They do not need hard shells because nothing is lining them up from across the shallows. Every measured feature of the Ediacaran biota fits this picture. sessile or slow-moving habits. No armor, no offensive weaponry, no evidence of predator-prey encounters. Even the deeper Precambrian record and the microbial mats that dominated the seafloor for 2 billion years before the Ediacaran shows the same pattern. Grazing on those mats where it happened was slow. Herbivorous interactions where they existed were gentle. The whole system ran on slow chemistry. This is what the world looked like, everywhere on Earth, for almost the entire history of life before the base of the Cambrian. A chemical ocean, a tactile ocean, an ocean in which distance was invisible, in which shadow carried no meaning, in which an organism could pass within centimeters of another and neither be aware of the other's existence until they touched. That was the longest-running state of complex life the Earth has ever held, more than 3 billion years. In a brief interval, geologists can still pick out of the sediment record with a few million years of precision, it was over. The first sign that something was changing is in the rocks, at the transition between the Ediacaran and the Cambrian, as very small hard parts begin to appear. They are called, collectively, the small Shelly fauna, sometimes abbreviated to SSF. They are not the beautiful, complete, articulated Cambrian animals we will meet a few million years later in the Burgess Shale. They are fragments, chips, scales, tiny conical shells the size of grains of rice, small tubular structures of calcium carbonate or calcium phosphate, isolated hard pieces that clearly once belonged to something larger, because the soft parts of that something did not fossilize, appear now as isolated shapes. The small Shelly fauna begins to appear in the latest Ediacaran in small numbers, and then, across a span of roughly 10 to 15 million years at the base of the Cambrian, it increases in diversity and abundance until the rocks are full of these tiny, hard structures. By about 520 million years ago, most of the world's carbonate shelves are carpeted with SSF fragments. For the first time in Earth's history, is producing biologically made hard parts in large quantity and variety. Why now? Part of the answer is chemical. The late Neoproterozoic and earliest Cambrian show a pulse of atmospheric and oceanic oxygenation, one of the later steps of the Neoproterozoic oxygenation event that appears to cross a threshold supporting energetically expensive biomineralization. Making calcium carbonate shells, and especially the more metabolically costly phosphate skeletons, requires sustained energy investment from the cellular machinery of the organism. Low-energy organisms cannot do it. High-oxygen, high-energy organisms can. The oceans of the latest Precambrian had finally reached the metabolic regime where hard parts became energetically possible. But oxygen alone does not explain why. Earlier pulses of Proterozoic oxygenation had not triggered a biomineralization wave. Something else was operating at this particular time. Something that made the construction of hard parts not merely possible, but selectively advantageous. Something that made the organisms that invested in shells and spines, sclerites, outcompete those that did not. The leading hypothesis, the one we will spend much of this story building toward, is that the selective pressure was visual, that the arrival of eyes somewhere in the early Cambrian ecosystem for the first time in Earth's history, an environment in which predators could systematically target soft-bodied prey from a distance, and in which evolving hard parts were worth the metabolic cost. But to understand why that is the hypothesis, we have to look more carefully at what an eye is, how eyes work, in the stratigraphic record of the early Cambrian. the first one appears? The answer, once you know where to look, is preserved in stone. The earliest eyes in the fossil record belong to trilobites. This is a remarkable fact, and worth sitting with for a moment. Trilobites, the three-lobed segmented arthropods that dominated the Paleozoic seafloor for more than 250 million years, appear in the rock record at around 521 million years ago. When they first appear, they already have eyes, not light-sensitive patches, not precursor dimples, full, functional, image-forming compound eyes with visible lens arrays built into the front of their carapaces. The fossils preserved them. Holding a well-preserved trilobite in your hand today, look directly at the faceted surface of the animal's eye, in exactly the position it held when the creature was alive 500 million years ago. The preservation is possible because of the specific mineralogy of the trilobite eye. Trilobite eye lenses were made of a single crystal of calcium carbonate, grown in a precisely controlled crystallographic orientation. This is, in itself, an extraordinary biological feat. Calcite, in its natural state, has a property called birefringence. It splits incoming light into two beams with different polarizations, bending each one to a different angle. This is the kind of behavior that makes calcite a useful material for optical research, but a terrible material for an eye. A lens made of randomly oriented calcite would produce a double image, blurred and unfocused, useless for vision. Trilobites solved the problem by growing their lenses with the optical axis of the calcite crystal aligned precisely along the direction of incoming light. In this orientation, known as the C-axis of the crystal, the birefringence effect collapses and the calcite transmits light essentially without doubling. Every lens in a trilobite's compound eye was a carefully grown single crystal, oriented so that its optical properties were invisible to the creature looking through it. This means the trilobite eye, when you study it in the fossil, is not a soft tissue impression or a vague trace. It is the original optical apparatus, preserved in its original mineral. Among all the organs of all the animals that have ever lived on Earth, trilobite eyes are perhaps the best preserved, sometimes millimeter perfect, sit in the rock exactly as they sat on the living animal. The oldest of them is roughly 500 million years old. And when you examine them optically, when modern paleontologists have reconstructed the geometry of the lens arrays and calculated how light would have traveled through them, the picture that emerges is astonishing. The trilobite eye, in its earliest forms, was already a sophisticated imaging device, not a primitive attempt, not a precursor stage, a fully functional compound eye with measurable resolving power, with a defined field of view, with properties comparable in principle to those of modern arthropod eyes. Some of the later trilobite eye designs are even stranger. Certain Devonian trilobites, like the genus Dalmatitina, evolved lenses with a specific doublet shape, a bulged upper portion and a concave lower portion that corrects for spherical aberration using exactly the same optical principle that modern camera lens designers use in their multi-element achromatic corrections. The trilobite lens, in these later forms, is a pre-human achromatic design grown biologically 360 million years before the first human optician ever ground a piece of glass. The point of this is not merely to admire the trilobite, though the trilobite is worth admiring. The point is that the eye, in its first appearance in the fossil record, is not simple. It is already sophisticated, which means the evolutionary work of building it had already been going on in lineages whose soft tissues did not fossilize for some significant time before the earliest trilobite we can find. The eye had been under development for long enough that, by the time hard parts appear to preserve it, it is already a finished instrument. To understand how a compound eye works, you have to understand that it is not one eye. It is a collection of many small eyes, each with its own lens, its own photoreceptor, its own narrow field of view. Each individual unit is called an ommatidium. An ommatidium, by itself, sees only the direction in which it points. The information it returns is, in effect, a single pixel. Multiple ommatidia, packed together across the curved surface of the eye, each pointing in a slightly different direction, return many pixels simultaneously. The animal's nervous system combines these pixels into a single mental image, assembled across the whole eye at once. The resolution of a compound eye is determined by the number of ommatidia and the angular separation between them. More ommatidia covering the same field of view means finer resolution. More widely spaced ommatidia covering a wider field means broader coverage but lower detail. Insects and other arthropods have spent the last half-billion years evolving different solutions to this trade-off, adapted to different ecological needs. A dragonfly, for example, has about 28,000 ommatidia per eye, an enormous number, giving it the acute motion detection it needs for hunting other insects in flight. A worker ant might have only a few hundred. A crab, adapted to wider field monitoring of its environment, has relatively few but widely spaced units. The underlying architecture is the same. The resolution varies by species and by need. The earliest trilobite eyes had roughly 100 to 300 ommatidia. Modest by modern standards, but not trivial. A 300-lens compound eye, in a creature a few centimeters long, gives enough visual resolution to detect movement in the water, to recognize the shape of a nearby object, to track the approach of something larger. It is an eye fully capable of behavioral use. It is not ornamental. It is not a byproduct of some other organ. It is, from the moment of its first appearance, doing visual work. And then, in the Emu Bay Shale of South Australia, a different creature was found, a creature not a trilobite, not even closely related to a trilobite, but contemporary with them, whose preserved eye is, in optical terms, considerably more advanced. The creature is called Anomalocaris, or more precisely, because the name has been applied over the years to several related species, and the taxonomy has been complicated by the initial confusion about what the creature even was. The creature is a large radiodont, a member of the group of early Cambrian predators that dominated the apex positions in the food chains of the time. Anomalocaris, at its largest, was perhaps 1 meter long. By the standards of the early Cambrian, when most animals were a few centimeters long at most, this was enormous. A meter-long body, driven through the water by undulating lateral flaps arranged along its flanks, with a round-toothed mouth underneath and 2 large articulated frontal appendages reaching out from its head like grasping arms. The arms, carrying backwards curved spines, were designed to clamp onto prey and draw it into the mouth. A ring of hard plates that could open and close like a constricting iris was lined with backwards-facing hooks for drawing food inward. And above the mouth, on 2 short stalks projecting from the front of the head, sat 2 spherical compound eyes. The eyes of Anomalocaris were first recognized for what they were in a 2011 paper published by a team led by paleontologist John Patterson, working on specimens from the Emu Bay Shale in South Australia. The preservation at Emu Bay is exceptional. Thin sheets of shale that preserve fine detail, including the faceted surfaces of compound eyes, with enough clarity that individual lenses can be counted. And when the Patterson team counted them, the number was extraordinary. 16,000. 16,000 individual lenses in a single Anomalocaris eye. Each lens, independently transparent, arranged across the curved surface of the eye in a precise array. The total complexity of the eye put it in the same league as modern dragonflies. Anomalocaris, 500 million years ago, was carrying around visual hardware that was, in optical terms, essentially competitive with the best of what modern arthropods have developed after another 500 million years of refinement. What this means, ecologically, is clear. Anomalocaris was an active pursuit predator. Its eyes would have given it the ability to spot prey at significant distances in clear sea, to track that prey as it moved, to maneuver its body toward an interception point, to strike with its grasping arms when the prey was within reach. Its hunting style, in other words, was essentially the hunting style of a modern predator. Visual targeting, calculated pursuit, precise strike. Something no creature on Earth had been able to do in any substantial way before it evolved. The discovery of the Anomalocaris eye is, in a sense, the exact moment at which the story of Cambrian predation became visible. It is one thing to infer from indirect evidence that Cambrian predators must have been using vision. It is another to see directly the optical hardware preserved in stone. The Emu Bay specimens push the case from hypothesis to observation. And once you start looking, the Cambrian fossil record is full of eyes. Opabinia regalis was smaller than Anomalocaris, perhaps 7 or 8 centimeters long, and considerably stranger. Its body was segmented, flattened, with rows of lateral swimming flaps running down each side. Its head was broad and carried as its most distinctive feature, 5 eyes. Arranged in a cluster across the top of the head, each one mounted on its own short stalk, each one independently oriented. The central 3 eyes faced forward and slightly upward. The 2 outer eyes faced more to the sides. Together, they provided, as best as we can reconstruct it, an almost complete dome of visual coverage. Opabinia could see above, ahead, and to either side all at once without needing to turn its head. In front of the eyes, extending from the base of the head, was a long, flexible proboscis ending in a grasping claw. The proboscis could be extended forward, used to grasp small prey, and then bent back underneath the head to bring the captured food mouth on the underside of the body. When it was first described from Burgess Shale specimens in the 1970s by the paleontologist Harry Whittington, produced famously outright laughter at the conference where he presented the reconstruction. 5-eyed predators with grasping hoses were not the standard picture of early Cambrian animals that the audience was used to, things anyone had expected. But the fossils were unambiguous. The eyes were there. The proboscis was there. Opabinia was real, and it had been real. And it had lived and hunted in a Cambrian Sea 508 million years ago. It is one of a large group of strange, early arthropod-like creatures that, across the Chengjiang and Burgess Shale and Sirius Pisset and Emu Bay faunas, represent the first great experiment in visual predation. Lirarapax, Amplictobolua, Hurdia, the radiodont cousins of Anomalocaris, each with their own variations on the basic body plan, each with compound eyes of their own. A smaller arthropod with multi-fin grasping limbs and large forward-facing eyes. Carigmacula from Greenland, a lobopod-like creature with two large frontal appendages and prominent eye stalks. The Cambrian oceans, within a few tens of millions of years of their beginning, had become populated by a carnival of visually equipped predators, each one experimenting with a different combination of eyes, body plan, and hunting strategy. It was, for the first time in the planet's history, an ocean in which to see was to survive. And those eyes, the Trilobite eyes, the Anomalocaris eyes, the Opabinia eyes, and all their cousins, even though they belonged to organisms that were, anatomically, very different from each other, all shared something in common at the deepest level of their genetic and cellular machinery. They all used the same master switch. In 1994, the Swiss developmental biologist Walter Gehring, working in Basel, performed one of the strangest and most quietly shocking experiments in modern biology. He took a gene out of a mouse. The gene, which had been studied for years, was known to control the early development of the mouse's eye. If the gene was damaged or missing, the mouse failed to develop eyes at all. If it was present and active, the eye developed normally. It was, in the standard language of developmental biology, a master control gene for eye formation, the gene at the top of a long cascade of other genes, the one whose activity triggered everything that followed. Gehring took that mouse gene and he inserted it into the embryo of a fruit fly, a completely different animal, evolutionarily separated from mammals by more than 600 million years of independent history. He inserted it in a specific location on the fly's body, where it would be expressed in cells that were not, under normal circumstances, going to develop into eyes. And when the fruit fly developed, extra eyes, structurally fly eyes, complete compound eyes with ommatidia, grew in the place where the mouse gene had been inserted. The eyes were fly eyes, because the cells that built them were fly cells, but they were built and triggered to be built by a gene that came out of a mouse. The gene is called PAX6, and it is one of the most conserved genes in animal biology. The conservation is not casual, it is extraordinary. The PAX6 sequence in a human, in a mouse, in a fly, in a squid, in a planarian worm, in a nematode, across all animals that have any kind of eye, is recognizably the same gene. The protein it produces is, in its key functional regions, almost identical across the entire animal kingdom. It has been conserved with remarkable fidelity for something approaching 600 million years, across lineages that otherwise differ in almost every other respect. What PAX6 does in every one of these animals is sit at the top of the eye development cascade. It activates a whole network of downstream genes that build lenses, photoreceptors, optic nerves, eye cups, and all the other components of an eye. The specific details of which downstream genes are activated depend on the species. A fly's PAX6 turns on fly-specific genes that build compound eyes. A mouse's PAX6 turns on mouse-specific genes that build camera eyes. But the master switch itself is shared. It is the same molecular trigger inherited from a common ancestor, conserved across every line of descent that led to a visual animal. The implication is clear. Every eye on Earth in every animal traces back to a single evolutionary origin at the base of the animal kingdom. PAX6 was present in the last common ancestor of all modern bilaterians. the group that includes arthropods, mollusks, vertebrates, virtually every animal with left-right symmetry. That ancestor almost certainly lived in the very late Ediacaran or earliest Cambrian. And it must have had, at minimum, some light-detecting structure whose development PAC6 was already coordinating. From that one ancestor, the gene was inherited by every animal that followed. The specific architecture of the eye, whether it would be a compound eye, a camera eye, a pinhole eye, a simple eye cup, would vary depending on the lineage. But the master control switch, the gene that said, "build an eye here," was already in place, and every animal that ever built an eye did so by flipping the same switch. This is part of why eye evolution in the Cambrian is, in a certain sense, not as difficult a problem as it might seem. The eye was not invented from scratch 50 separate times. It was developed, at least in its foundational regulatory circuitry, once, probably in a small, soft-bodied Ediacaran ancestor with at most a patch of photosensitive. And then, across the early Cambrian, that regulatory circuitry was elaborated into dozens of different forms, in dozens of different lineages, in response to a sudden and powerful selective pressure for visual information. What was that selective pressure? The Oxford evolutionary biologist Andrew Parker published a book called In the Blink of an Eye, in which he proposed what has since come to be called the light switch hypothesis for the Cambrian explosion. The hypothesis, in its simplest form, runs like this. The Cambrian explosion, the rapid diversification of animal body plans between roughly 535 and 520 million years ago, was not primarily triggered by a change in atmospheric oxygen, or a change in ocean chemistry, or a genetic innovation, or a climatic shift. It was primarily triggered by the invention of vision. Before vision, the ecological landscape was, as we have already seen, soft and slow. There was no distance predation, so no distance evasion. Animals were soft, unarmored, largely sessile, interacting at short range through chemistry and touch. An entire evolutionary equilibrium, stable for tens of millions of years across the Ediacaran, ran on those terms. The moment the first functional eye evolves, the equilibrium breaks. A predator with vision can now detect prey across a distance that was previously invisible. A prey animal without vision has no warning. The predator's advantage is asymmetric and devastating. Within a small number of generations, selection begins to act hard on every soft-bodied organism in the ecosystem, and the selection is multi-directional. For prey animals, evolve hard parts to make predation harder. Evolve spines to make yourself dangerous to swallow. Evolve camouflage to disrupt visual recognition. Evolve speed to flee after detection. Evolve burrowing to hide. Evolve eyes yourself so that you can see the predator coming. For predators, evolve even better vision. Evolve speed to intercept. Evolve grasping limbs to strike. Evolve size because larger predators can overpower more prey. Evolve multiple sets of grasping appendages. For everyone, evolve sensory integration. Develop nervous systems that can process visual information. Develop the brain circuitry needed to turn visual pixels into behavior. The consequence, across even a geologically short interval, is an enormous increase in the speed of evolution across the whole ecosystem. Every lineage is now under intense, sustained directional selection. Body plans are tested against predators and against each other. Most experiments fail, but the ones that succeed diversify rapidly because the selective pressure is so strong that small advantages sweep through populations very fast. The Cambrian Explosion, on this view, is the evolutionary signature of this single moment, the moment when vision became common enough in the ecosystem to remake every other interaction. Is the light switch hypothesis correct? Probably, in part. It cannot be the whole explanation because other factors, the pulse of oxygenation in the latest Ediacaran, the breakup of Rodinia, the innovations in genetic regulation that preceded the Cambrian, contributed in their own ways. But the timing of the appearance of the first eyes and the timing of the first widespread biomineralization and the timing of the first clear predator-prey interactions preserved in fossils all line up remarkably well with what the light switch hypothesis predicts. The eye does appear, based on the best current evidence, very early in the Cambrian sequence, and the rest of the Cambrian ecological transformation does appear in the rock record to follow it rather than precede it. Whatever the exact balance of causes, the effect is unmistakable. For 100 million years, atmospheric oxygen had been high enough to support complex animals. For tens of millions of years, the Ediacaran biota had lived in a world that could have supported biomediation, predation, arms races, but did not. Something changed at the base of the Cambrian that set the arms race in motion. The growing weight of evidence suggests that what changed, more than anything else, was that one of the creatures in the sea opened its eyes. The consequences of that opening are written in the fossil record across every continent. The small Shelly fauna, which we met earlier, is part of the signal. Appearing suddenly in the rocks at the base of the Cambrian represent an evolutionary response to a new predation pressure. Individual spines, scales, and shells, the first widespread biomineralized structures in Earth's history, appear in huge numbers in latest Ediacaran and earliest Cambrian sediments. But the harder evidence for an arms race is in the bite marks. By 510 million years ago, Cambrian fossil beds around the world preserve specimens with healed and unhealed wounds, trilobites with chunks missing from their thoracic segments, the edges of the wounds showing regrowth patterns that indicate the animal survived the attack, shells with partial drill holes, evidence that some predators were already developing the chemical or mechanical drilling behaviors that would become a hallmark of later gastropod predation. Burrow systems, a brand new phenomenon at this time, because burrowing did not exist in the Ediacaran, record the movement of prey animals seeking shelter in the sediment and of predators chasing them there. Burrowing, in fact, is one of the clearest signals of the Cambrian transformation. The substrate itself changes character at the Ediacaran-Cambrian boundary. Ediacaran seafloors were dominated by thin, laminated microbial mats because nothing disturbed them. Cambrian seafloors, within a few million years of the boundary, are dominated by mixed, churned, oxygenated sediment because animals have begun to burrow aggressively into them. This phenomenon, known as the Cambrian substrate revolution, records the death of the microbial mats that had dominated every sea floor on Earth for 2 billion years. They were, in effect, plowed under by the new animals. With burrowing came complex behavior. Trilobite trackways, called Cruziana and Russophycus in the trace fossil record, preserve the actual paths individual trilobites walked across the sea floor. Some of them record meandering feeding paths, showing that the animals were systematically scanning substrate for food. Others record straight-line flight paths, showing escape behavior. Others record resting depressions, where trilobites settled into the sediment overnight or between feedings. The complexity of behavior preserved in these trace fossils is orders of magnitude greater than anything the Ediacaran ever produced. The animals were not passively sitting, they were hunting, they were searching, they were fleeing. They were responding in real time to their environment. And increasingly, that environment included other animals who were watching them. Predation, in fact, seems to have moved very quickly through the ecosystem. The earliest small shelly fossils include conical tubes and rounded shells that were almost certainly the armor of small animals responding to predation. Within a few million years, these have been replaced or supplemented by more elaborate forms. Spiked sclerites, helical shells, jointed carapaces, integrated body armors. The classic Cambrian body plans, the trilobites, the brachiopods, the mollusks, the arthropods, all appear in the Middle Cambrian, already heavily armored. By the time of the Burgess Shale, 508 million years ago, the default condition for a Cambrian animal is to wear something. And the ones that did not, the ones that persisted with the soft, unarmored Ediacaran body plans, are not in the Cambrian fossil record. They had been eaten. The Burgess Shale is, by any standard, one of the most important fossil deposits ever discovered. It was found in a classic accident of fieldwork by the American paleontologist in British Columbia. Walcott, according to the often told story, was riding a horse with his wife along a trail when the horse stumbled on a loose slab of shale that had tumbled down from higher up the slope. Walcott dismounted, picked up the slab, examined it, and saw, observed in the rock, a creature unlike anything he had ever seen before. He returned the following year, located the outcrop from which the slab had fallen, and began the systematic collection that would eventually produce tens of thousands of specimens of early Cambrian animals. The Burgess Shale preserves the creatures of a Middle Cambrian seafloor, approximately 508 million years old, in extraordinary detail. The rocks are a fine gray-black shale, laid down in an oxygen-poor deep-water basin just off the edge of a carbonate platform. Creatures living on or near the platform were periodically swept off the edge by underwater mudflows, carried down into the anoxic deep water, and buried quickly enough that their soft tissues, normally the parts that never fossilize, were preserved. The result is a fossil assemblage that includes not only the hard shells and carapaces that usually dominate Cambrian deposits, but also the guts, the muscles, the gills, the eyes, the tentacles, the proboscises, the soft, fleshy bodies of creatures whose forms would otherwise be entirely unknown. The first great wave of interpretive work on the Burgess Shale was done by Walcott himself. He classified the animals he found into existing modern groups: arthropods, worms, mollusks, chordates, and the taxonomy he proposed stood for most of the 20th century. But in the 1970s, a second great wave of work, led by Harry Whittington at the University of Cambridge and students Derek Briggs and Simon Conway Morris, revisited the fossils in extraordinary detail, using new techniques of physical preparation to expose structures Walcott had not been able to see. What they found was that many of Walcott's original classifications were wrong. The Burgess fauna contained not just variations on modern body plans, but creatures that fit no modern phylum at all. Creatures with body architectures that had been tried once in the Cambrian and had subsequently gone entirely extinct. Anomalocaris was one of the results of this reinvestigation. For decades, the frontal arms of Anomalocaris, preserved as isolated specimens in the Burgess, had been interpreted as the bodies of shrimp-like creatures. The toothed mouth had been interpreted as a jellyfish. The main body had been interpreted as a sea cucumber. It took Whittington's team, working in the 1980s, to recognize that these three apparently separate organisms were, in fact, parts of a single predator. A meter-long radiodont that had been misidentified as three smaller unrelated animals for decades. Opabinia, similarly, was redescribed. A creature whose original reconstruction had shown it walking on its spines with tentacles waving above was, through later work, turned upside down. The tentacles became legs, the spines became defensive structures on its back. Wiwaxia, a small creature covered in overlapping sclerids, emerged as an early relative of mollusks and annelids. Morella, an elegantly spined small arthropod, turned out to be the most abundant animal in the entire Burgess fauna, with thousands of specimens collected. The picture that emerged from the Burgess Shale was, and remains, the single best snapshot of a Cambrian ecosystem that we possess. A shallow reef-edge community, 508 million years ago, with predators hunting visually, prey armored and spined, burrowers in the sediment, filter feeders on the substrate, and a range of body plans that included everything modern phyla descend from, and a great many that did not survive. The Burgess Shale, more than any other fossil deposit, is what the early Cambrian looked like. But the Burgess Shale is not the earliest such deposit, nor is it the oldest window into the Cambrian we now possess. In 1984, near the village of Maotianshan in Yunnan Province, southern China, paleontologists investigating unusual black shales found, preserved in the rock, animals very much like those of the Burgess Shale, but older. The Chengjiang biota, as it came to be called, dates to roughly 518 million years ago, a full 10 million years older than the Burgess. And the quality of preservation is, if anything, even more remarkable. The shales preserve soft parts in such detail that eye facets, muscle fibers, gut contents, and nervous system traces can be recovered. Over the past 40 years, tens of thousands of specimens have been collected from the Chengjiang sites, and they have transformed our understanding of early Cambrian life. The Chengjiang fauna, being 10 million years earlier, preserves the Cambrian ecosystem closer to its origin. Many of the same basic body plans are present: radiodonts, lobopods, trilobite ancestors, early chordate, but in more primitive form than their Burgess cousins. The Chengjiang Seas were shallower, probably warmer, and preserved a slightly different assemblage, with more delicate creatures and with representatives of groups not yet present in the later Burgess. One of the most important Chengjiang finds is Hycoichthys ercaicunensis. It is a small creature, only a few centimeters long, preserved as a slender dark impression on the pale rock. And it is, as far as we can tell, the oldest unambiguous vertebrate, the oldest member of the lineage that will eventually, across more than 500 million years, produce you. Hycoichthys has a notochord, the proto-spine of early chordates. It has a well-defined head, with traces of what appear to be eye capsules. It has gill structures. It has segmented muscle blocks running along its body. It does not yet have a proper skull or jaws or limbs. But it is, unambiguously, a fish-like animal, with the body plan that all vertebrates will inherit. And even this earliest vertebrate appears to have had eyes, small, pigmented paired eye structures at the front of its head. It is worth sitting with this for a moment. The oldest vertebrate we know, at 518 million years ago, already had eyes, which means the vertebrate lineage, from its very origin, was a visual lineage. We did not acquire eyes later or independently. We inherited them from the common stock of early Cambrian animals, already equipped. The Chengjiang also preserves Milocunmingia, a similar early chordate, and various other creatures that fill out the picture of early vertebrate origins. These animals lived in ecosystems dominated by Anomalocaris and its radiodont cousins, in seas where large visual predators were already the top of the food web. The first vertebrates were prey animals. They were small, fast, and already carrying, in their forward-facing eyes, the sensory equipment that would carry their descendants across the entire subsequent history of life on Earth. A question that often comes up at this point is, how long does it take to evolve an eye? The question seems, at first hearing, as though it might not have an answer. Eyes are complicated organs involving lenses, photoreceptors, nerve connections, and intricate developmental pathways. Surely one might think such a structure must take tens or hundreds of millions of years to evolve. The Swedish biologists Dan Erik Nilsson and Suzanne Pelger set out to calculate exactly this using a formal mathematical model. They began with a flat patch of photosensitive cells, the absolute simplest possible starting point, a region of skin that could detect the difference between light and dark. They modeled small, incremental changes, a slight deepening of the patch into a cup, a slight narrowing of the cup's opening, a slight thickening of the covering membrane into a lens, a slight rounding of the lens for focus. For each change, they calculated the improvement in visual acuity, expressed as the percentage improvement in the resolving power of the organ. Using conservative assumptions about how quickly small evolutionary changes could accumulate under selection pressure, assuming that each step in the process produced only a modest improvement, and that the improvement had to spread through a breeding population before the next step could begin. Nielsen and Pelger calculated the total number of generations required to go from flat photoreceptor patch to fish-like camera eye, spherical focusing lens. The answer was approximately 364,000 generations. For a small marine animal with an annual breeding cycle, that is roughly 364,000 years, less than 500,000 years. For an animal with a faster generation time, many early Cambrian animals probably bred more than once a year. It could be considerably less. Let that sit for a moment. From a patch of light-sensitive cells to a fully functional camera eye, under ordinary evolutionary conditions, in less than 500,000 years. The entire transformation that intuitively seems as though it would require geological time can in fact be accomplished in an interval so short that, in the stratigraphic record, it would barely register as a single rock layer. This calculation changed the way many biologists thought about the Cambrian eye. Once it was established that the evolution of an eye, starting from a precursor photoreceptor, is a fast process on geological timescales, the question of the Cambrian became not how could eyes have evolved that quickly, but how could eyes not have evolved once the ecological and genetic preconditions were in place. The light switch hypothesis received, quietly, an important piece of supporting mathematics. And the corollary is important too. Eyes, once they evolved in one lineage, could and did evolve independently in many others. The camera eyes of vertebrates, the compound eyes of arthropods, the camera eyes of cephalopod mollusks. Each of these represents a separate elaboration along somewhat different developmental pathways. from a shared Pax 6-driven starting point. The underlying genetic switch was the same. The architectures built on top of that switch differed, and all of them could be built under selection pressure in geologically brief intervals. By the Middle Cambrian then, the ocean had become a seeing ocean. Predators hunted visually, prey fled visually. Ecological interactions that had, a few tens of millions of years earlier, run entirely on chemistry and touch, now ran, in large part, on light. Body plans had diversified to accommodate the new sensory world. Defensive armor, offensive weapons, camouflage patterns, visual displays for mate recognition, and intraspecific competition, all of these had appeared in some form in the oceans of the Middle Cambrian. And the changes would continue across the next 500 million years in every animal lineage that emerged from the Cambrian stem. The vertebrates, starting with Hyquictus and its Chengjiang relatives, would refine their camera eyes across 400 million years, producing the sharp-focused eyes of fish, the lateral eyes of reptiles, the stereoscopic eyes of primates, the ultraviolet detecting eyes of certain birds and insects. The arthropod line, starting from the compound eyes of trilobites and radiodonts, would produce the facet-dense eyes of dragonflies, the motion-sensitive eyes of mantis shrimp, the bifocal stalked eyes of certain spiders. The mollusks, entering the Cambrian with simple eyes, would ultimately produce the sophisticated camera eyes of cephalopods, eyes so similar to vertebrate camera eyes in structure and in optical performance that their independent evolution from a shared Pax 6-driven starting point is one of the clearest examples in biology of convergent design. Each of these visual lineages is, in a real sense, a direct continuation of the Cambrian invention. The specific eyes have changed, the basic grammar has not. Across 600 million years, no animal lineage has fundamentally reinvented vision. They have only refined what was worked out in those early, strange Cambrian seas. The genes, most of them, are the same. Pax 6 is still there. Opsins, the photoreceptor proteins that actually capture photons, are still there in variations that trace back to a set of ancestral molecules present in the earliest Cambrian eyes. The developmental circuitry that turns a population of embryonic cells into a finished eye organ is, in its deepest logic, the same logic that was running in the Cambrian embryos that built the first eyes. This is not a metaphor. It is literal genetic inheritance maintained with remarkable fidelity across a span of time that is hard for a human mind to take in. The eye you have in your skull right now, inside the bone of your orbit, wiring light signals into the visual cortex of your brain, is built by a developmental program whose core regulatory logic is older than every mountain range, every ocean basin, every continent you have ever seen. It is older than almost anything you would call ancient. When you look at something, when you hold your gaze on a face or a horizon or a page of text, you are activating a system that was prototyped in a Cambrian ocean 520 million years ago, and that has been elaborated, but not fundamentally replaced, ever since. It is worth, before the story ends, trying to sit for a moment with what changed. For 3 billion years, the overwhelming majority of the history of life on Earth, nothing on this planet could see anything. The oceans were full of life. Microbial mats covered every shallow sea floor. Photosynthesis had been running for 2 billion years, pouring oxygen into the atmosphere. Eukaryotic cells had existed for more than 1 billion years. Multicellular animals in the form of the Ediacaran biota had appeared and flourished. But none of it, none of the microbes, none of the proto-animals, none of the frond-shaped Ediacaran creatures could look at anything. Light, in a planetary sense, carried no information. It was only light. In a span of time that may have been only a few million years, that stopped being true. Somewhere in an early Cambrian Sea, a population of small, soft-bodied predators or proto-predators, the first arrangement of cells appeared that could convert an incoming pattern of light into a corresponding pattern of nerve impulses. At first, that pattern would have been crude, a dark shape moving, a bright spot against a dim background, nothing like the detailed visual world a modern animal sees. But it was, for the first time in the history of the planet, information. And once the first animal could see, the rest of the biosphere had to respond. The Cambrian Explosion, the remarkable radiation of animal body plans, the appearance of hard parts, the evolution of burrowing, the rise of complex behavior, followed in large part from that single innovation. Not because vision was magical, but because vision was, in an ecosystem that had never had it, a previously unavailable kind of information, and any lineage that could tap into it or respond to it had enormous evolutionary room to grow. We are, all of us, the long downstream consequences of that moment. The bird you hear calling from a tree this morning is looking at the world through an eye descended from the Cambrian. The cat that watches you from a windowsill sees through the same inheritance. The ants walking along a kitchen counter, each carrying inside its tiny compound eye a direct descendant of the Opabinia architecture of early trilobites, are participants in the same lineage. Every animal alive today, every mammal, every fish, every insect, every mollusk, all of them trace their ability to see through a chain of descent that passes, without interruption, through the early Cambrian seas. You, reading these words, or hearing them, if someone is reading them to you, are doing so through an eye whose master development gene has not changed in its core function, since long before the vertebrate lineage existed as a recognizable thing. Your eye is an heirloom. The seeing you are doing right now, however casually, is the continuation of an unbroken lineage of seeing that began on the floor of a Cambrian sea more than 500 million years ago. The world, before the invention of eyes, was not dark. There was sun. There was light. Photons reached the surface of the ocean and penetrated through the upper water. And they reached the skin and the bodies of living things, and they produced the same physical effects they produce today. It is only that nothing could turn them into meaning. And then, one morning in a Cambrian sea, something could. After that morning, the world had light the way we know light. It had images, it had distance. It had the shapes of other creatures, approaching or retreating. It had the possibility of tracking, of evading, of recognizing, of attending. It had the precondition for every sighted behavior that would ever be performed, from the first trilobite swerving to avoid an Anomalocaris to a human looking up at the night sky and seeing stars. The Cambrian Explosion is, among other things, the name for this change. When you next find yourself paying attention to a sight, a bird on a branch, a face across a room, the slow turn of leaves in a low wind, it is worth, just for a moment, remembering where the attending came from. Not a human invention, not a mammalian refinement. An inheritance from a population of small, soft creatures 500 million years ago, whose ancestors had existed for 3 billion years in the dark, for reasons that had as much to do with the availability of oxygen, the mechanics of PAC-6 expression as with anything else, opened a new kind of window on the world. We live now in the long afternoon of that opening. The world remains as it has been for roughly 520 million years, a world that can see itself. And the work of all those early small creatures, every trilobite, every Anomalocaris, every early chordate with its first small paired light-sensing spots, still lives in every glance of every seeing thing. It is a very old inheritance. It is, in a sense, the oldest inheritance that any conscious being has. The world, which we often forget is old, is still here, still seen, still being looked at by the long descendants of a Cambrian Sea. And every time any of us opens our eyes, we participate in a moment that began very quietly, a long time ago.